Today I'm going to be talking about Digenean life cycles and one of my favorite parts about Digenean life cycles, which is the social organization that the larval stages exhibit. These parasites in general have a 3-host life cycle. Although there are some variations, some might only have 2 hosts, some have up to 4 hosts, but in general, we'll talk about the generalized trematode life cycle. They have 3 hosts. So we start with the vertebrates. So for example, here we have some birds. Don't make too much fun of my drawings. In the vertebrate host, as I said, we have the adult Digenean. It reproduces sexually inside the vertebrate host, and the eggs are released out with the feces. They fall into the water, and out of the eggs hatches the myricydium. The myricydium has a lifespan of maybe about 2 hours. During that time, it's swimming around searching for the molluscan host, a snail. Once it finds the snail, it goes into the snail, it metamorphoses into a mother sporocyst, and then this mother sporocyst starts asexually producing either sporocysts or rhedia. So let me write here, sporocyst or rhedia. So the difference between these two is that the sporocyst is essentially a bag, and it has the next stage in the life cycle developing inside of it. But it doesn't have any other structures like a mouth part or a pharynx, gut, etc. The rhedia, on the other hand, does have a mouth part. It has a very muscular pharynx, and it has a blind gut. So it can use this pharynx, this mouth part, to prey either on snail tissue or another parasite, as I'll talk about in a little bit. So sporocysts and rhedi produce more sporocysts and rhedi, and then at some point they switch to production of the next stage in the life cycle, the cercaria. The cercaria will then swim out of the snail. It's got a lifespan of maybe 12 hours to 24 hours, and it'll be swimming around trying to find the second intermediate host. Once it finds the second intermediate host, it'll insist on or in that host. My favorite second intermediate host is the California killifish. There's this one parasite, which I call UHA for short. It insists close to its brain, and from there, it kind of takes control of the steering wheel. It can release certain hormones, neurotransmitters, chemicals that will control the behavior of the fish so that instead of swimming kind of low in the water where it's safe, it'll start swimming closer to the surface of the water and making flashy movements, essentially yelling at the bird, eat me, because the parasite ultimately wants to end up in the bird's intestine where it can reproduce sexually. Okay, so as we've seen, the trematode life cycle is quite complex. It has to use 3 different hosts in order to complete the life cycle. They can't skip any of these hosts. Reproduction takes place in 2 different parts of the life cycle. In the definitive host is where sexual reproduction takes place, but asexual reproduction is also happening inside the first intermediate host, the snail. It's because of these kind of 2 phases of reproduction that Digeneans receive their name. Di means 2, genesis, origins, so 2 origins because they are reproducing in 2 different parts of their life cycle. Okay. So this is what I wanted to cover about trematode life cycles. Now I'm going to talk a little bit about the social organization part. So whether you believe it or not, trematodes form colonies inside their first intermediate host, similar to maybe what you can think about it, ants, termites, bees, etc., where we have a colony that's working together to get food, to help the queen reproduce, etc. They have a reproductive queen and they have sterile workers. Well, it turns out these parasites have something similar. And we're going to talk a little bit about how we discovered this. So the first thing we notice when we dissect a snail is that about 40% of the snail's wet tissue is parasites. So these sporocysts or rhedia are just covering the snail. They're everywhere. The snail has been castrated and all the reproductive energy of the snail is just redirected towards producing more and more parasites. The snail is now like a parasite-producing factory. At the beginning of my PhD, I started looking carefully at these dissections. So I would dissect a snail, and I would see thousands of parasites spilling out, right? 40% of the snail's tissue was just parasites. And I noticed that about half of the parasites that spilled out were really large. Maybe they were orange, and they were filled with the next stage, the cercaria, over here. However, there were also a lot of smaller parasites that didn't have anything developing inside of them, but still had a huge mouth part. And they were just found in the snail in really high numbers. And there was nothing really in between those two sizes. So what I asked was, thinking about what I've seen with ants, bees, the dimorphism between soldiers and reproductives, could it be that these parasites had some sort of reproductive division of labor? And so I decided to study this for my PhD thesis. I will be telling you about the experiments I did and the results I found in a little bit, but first I'd like you to pause the video for a little while and think, if you were to address this question, how would you do it? What would you measure? What experiments would you set up? How would you go around asking this question? Reproductive division of labor does occur in these parasites, in these parasitic flatworms that infect snails and form colonies inside of snails. They form a colony because these are all individuals that are working very closely together to maintain control of their resources and exploit them as best as they can. They are defending those resources from invasion by other parasites. And then we have two very different morphs, where one, the reproductives, are specialized for reproduction, as we can see, because they have developing offspring inside of them that will then leave the parasite and the snail to continue on with the life cycle. But also we have many, many, many smaller sterile individuals that have really big mouth parts compared to the size of their body. These small ones are very common in the front of the snail, where we would expect invasions to occur. And also, they attack at much higher rates than their reproductive counterparts do. So that's all I have for you today. So if you have any questions about either trematode life cycles or the reproductive division of labor that we've found in some species of trematodes, please contact me. Thank you.